So let's think about the process of phagocytosis. We're going to focus on the process of phagocytosis in neutrophils first. So this diagram here, this big blue cell here represents our neutrophil. We know it's a neutrophil because it's got this multi-lobed nucleus, and we can see the presence of lots of lysosomes within the cytoplasm of that neutrophil. If our pathogenic microorganism gets past the physical barriers of the body, or the mechanical barriers, or the chemical barriers, if it enters into the body tissue fluids or lymph fluids or blood, then the neutrophil will be attracted towards that pathogen, and it will move towards the pathogen. It's usually attracted to chemicals that the bacteria are producing, so the movement of the neutrophil towards the pathogenic bacteria is described as chemotaxis. Taxis meaning to move, and chemo meaning chemical. This is a nice key word that will often get you extra credit in an exam answer. Once our neutrophil has moved by chemotaxis towards our pathogen, then the antigens that are on the surface of the pathogen will then bind to receptors that are found on the cell surface membrane of our neutrophil. And then our cell surface membrane of our neutrophil will start to engulf the bacteria. Now, we've looked at this process in semester 1, this process of endocytosis, and we can describe the neutrophil membrane as infolding around the pathogen or invaginating around the pathogen. And these projections here on our neutrophil, as that membrane infolds or invaginates, are called pseudopodia. What happens is when these pseudopodia meet, they will pinch off and they will form a membrane around the pathogenic bacteria, and this structure is called a phagosome, which are the bacteria encased within a membrane structure. What will then happen is the lysosomes that contain lots of hydrolytic enzymes, or you might see the hydrolytic enzymes described as lysosome, or you might see the hydrolytic enzymes described as lysins. These lysosomes will move and fuse with the phagosome, and they will release their hydrolytic enzymes into the phagosome, and those hydrolytic enzymes will destroy the bacteria. They will fully digest it. And when the bacteria have been fully digested, then the phagosome with the digested bacteria in will move to the neutrophil cell surface membrane and will release the digested contents from the cell in the process of exocytosis. Remember, exocytosis is a bulk transport mechanism, so we require ATP and therefore mitochondria for this process to take place. So this is the process of phagocytosis in neutrophils. We end up fully digesting the pathogenic bacteria. The attached PowerPoint has got a really nice little video that you can click on to watch to see that process taking place. If we think about the process of phagocytosis in a macrophage, then in a phagocyte that's slightly bigger and has more of a traditional looking nucleus, so it doesn't have the multi-lobed nucleus, this structure here represents our pathogen, with our red triangles on its cell surface representing the pathogen antigens. The pathogen will be engulfed by the macrophage in the same way as the pathogen was engulfed by the neutrophil. So we will have the cell surface membrane of our macrophage invaginating around the pathogen and enclosing the pathogen within a phagosome. It's what happens next that's different in a macrophage. So rather than the enclosed bacteria being fully digested, in our macrophages, the macrophage basically only partially digests the bacteria, and it cuts up the pathogen using the hydrolytic enzymes, but it keeps the antigens intact. And these intact antigens are then moved to the surface of the macrophage white blood cell itself, and the macrophage displays those antigens on its surface. So this is our macrophage white blood cell, but it has bacterial or pathogenic antigens on its surface. And the macrophage is now described as an antigen presenting cell. And we will look tomorrow at what these antigens are presented to and what further immune response will take place.